Good, good. Okay. They really turned that on, didn't they? I'm gonna move that there. Okay, is that about right? Yes, excellent, thank you. Okay, we're gonna go ahead and get started. Thanks everybody for coming. I know this is sort of the getting to the end of the last day of the conference, so thanks for sticking with us and attending this session. So this session is Spatial Transcriptomics Advances and Applications. My name is Amanda Poholek. I'm gonna be one of the speakers, and then we're gonna have two other speakers. I set this up to be kind of a more traditional style, so we will have 20-minute talks from each speaker and then sort of 5 minutes of Q&A at the end of each of the speakers. And feel free to come up and use the microphones in the center of the room if you have any questions. Okay, all good? All right, so I can keep on time. Okay, that microphone's a little easier. Okay, great. So as I mentioned, my name is Amanda Poholek. I'm an associate professor of immunology at the University of Pittsburgh. And I also am the director of the Health Sciences Sequencing Core at the University of Pittsburgh School of Medicine. And so I decided to structure my talk to start a little bit about the core facility and how we are doing spatial transcriptomics in the core. And then I'm going to share a little bit about the science that I do in my lab, in particular highlighting some of the spatial technologies that we've used, and in particular focusing on some of the computational approaches that we have developed in collaboration with computational immunologists in order to understand new biology using spatial. So as I mentioned, I'm the director of the Health Sciences Sequencing Core, and so we traditionally provide sequencing services for most of the University of Pittsburgh and occasionally some external customers. And around about 2019 or so, as we started to hear more and more about spatial transcriptomics, it became clear that this was something that as a core facility we wanted to be sort of at the leading edge in being able to offer. And so with that in mind, we started first performing some of the spatial transcriptomics in my lab so that we knew that we could do them successfully, and then that gave us the ability to then offer those services as a core facility. And I think we did a pretty good job of jumping on this on the early side, where we were able to launch the sort of the V1 Visium from 10x as early as January 2022. And then as the technologies advanced with Curio Slide Seeker coming out in 2023, and then finally transitioning to the Visium HD and Xenium in 2024. So we're now able to offer all of these platforms in our facility, although we're now primarily mostly doing Visium HD and Xenium. And of course, the core is nothing without its staff. So these are the people, all three of these people are at this meeting. Hopefully many of you have had a chance to interact with them. If you haven't, hopefully you still have a little time. So Will McDonald was sort of really helpful at the beginning of getting our core up and running for spatial, and then we were very fortunate to recruit Yvonne, who is here in the room, who had expertise in histology and in protein, which really helped sort of put those two pieces of spatial together, the histology component together with the sequencing component. And then our senior technologist, Rania, who had been with our core actually longer than any of our staff, she was able to sort of come into the spatial group as we sort of got things off the ground and moved things forward. I think Rania's also here today as well. So these three people are really the people who go every day in and out to manage the components of spatial that can become challenging, learn all the tricks, and figure out how to offer this successfully as a core service to the community. And one of the things that was important to me as a core director when we got started on this was to be able to offer this to our customers in sort of an end-to-end solution process. And so our current workflow that we've grown to be fairly comfortable with is as follows, where we usually start when someone contacts us with an interest in doing spatial, and we try to meet with them either in person or by Zoom within less than 1 week. And this really is a key component to understand the individual project pieces that will be important for understanding how to do this successfully. And I say that because I'm sure as many of you either do spatial or have thought about doing spatial, you probably are aware, every tissue, every project, every individual situation has its own unique little thing that makes it a challenge. And so really digging in with our customers at this early stage to understand exactly what their needs are, what they're looking to accomplish, and what the particular challenges of their project might be really helps us to do this successfully. Once those samples are then delivered to us, we're able to sort of begin the actual wet bench project portion of that. And for us, a key component of this is to establish a really strong relationship with a histology core that's located nearby to us. And so in that relationship, we're really able to get the samples cut and prepared in the way that we want them onto the slides, do RNA quality checks, and then report back to the customer whether those RNA quality checks are good or not. And once those samples pass QC, we can then move on to spatial assays, depending on whether they're doing xenium or visium. There's a lot that goes into that RNA quality check. I could do a whole 20-minute talk just on that. So I'm not going to do that today because I think most of you probably know the challenges with that. And then at the very end, we do offer the data delivery. One portion that we haven't offered in terms of this end-to-end solution is the complex analysis portion. We'll do the basic primary analysis, but we have not done more than that. We've been offloading that to bioinformatics cores at the University of Pittsburgh who have more experience than us. So by and large, this has been still a very successful service. This slide is actually not even fully updated, and so we probably have done closer to 50 projects and probably over 350 samples across many, many different human and mouse tissues. And so we're very pleased that this has resulted now that we're 4 years in publications and in many publications from different groups across the University of Pittsburgh. So this is one of our earlier ones using the V1 Visium, looking at spatial of scleroderma skin lesions in pediatric patients. So as you can imagine, many clinicians have been very quick to adopt spatial transcriptomics, especially those that are already sort of pathology oriented. More recently, we've had some of the work that we've done be published in a study looking at 2 different immunotherapies combined in advanced solid tumors. And then we have recently 3, as we've transitioned to Visium HD, 3 groups that have preprints out that are using the Visium HD to both understand biology as well as generate new computational methods, including Zeitgeist, which is looking at how to deconvolve spatial transcriptomics data, DGAT, which is also a new spatial protein inference computational approach, and then finally another clinical group looking at liver transplant rejection and looking at the potential drug targets that can be found there. So that's just a little bit about our core facility and how we've been able to provide spatial transcriptomics to the University of Pittsburgh. And if you have any specific questions about that as a service, I'm happy to answer those questions sort of after the talk. I can hang out for a little bit. I want to transition now to talking a little bit about how in my lab as a scientist, we've been using spatial to understand biology and into understanding and to using new computational methods to try and really fully flesh out the benefit of spatial. Because I think many of you probably also saw with single-cell transcriptomics, there was an early period of time where people were generating data, but how to actually analyze it was still a question. And until a lot of the computational approaches came along, we couldn't really fully dig into the biology. I think we're in the same place there with spatial, where we're sort of still developing many of the computational tools that are needed to really get at the biology that we can answer with spatial technologies. So in my lab, I'm interested in allergic asthma. I'm interested in how the immune response develops and how that ultimately leads to allergic asthma. And so allergic asthma, I'm sure, is something that many of you here are aware of. Almost probably everyone knows someone who has asthma. Asthma affects millions of people, both in adults and in children. And from the perspective of an immunologist, allergic asthma occurs when you breathe in something that someone normally would breathe in and not have a response to, but instead has an inflammatory response and begins to mount an inflammatory response that's detrimental to the lung and drive, obviously, the physical manifestations that you think of with asthma, wheezing and coughing and an inability to breathe. And it's really the immune system that underlies all of those components, where you have a series of different immune cells that directly produce cytokines that interact with the cells of your lung, including sort of the airway smooth muscle, the epithelial cells that produce mucus, and lead to dysfunction, fibrosis, scarring, constriction, and mucus production that leads to really what underlies those physical properties of allergic asthma. And so as an immunologist, we become very interested in understanding what are the individual components in the immune system that drive each of these processes, and more importantly, how does it get started? Why do you initially overcome tolerance and actually start to mount an immune response when most people do not? And so many decades of research have been done, and what we know is that a specific type of T cell called a type 2 or a Th2 T cell is critically important in this process and produces many of these type 2 cytokines, as shown here, that actively and directly provide many of these problematic issues for the epithelial cells and for the bronchial airway smooth muscle. So these type 2 cytokines like IL-13 and IL-5 coming from these type 2 T cells directly act on the lung physiology itself to produce all of these problems that actually drive allergic asthma. And we also know the antibody production can also occur in this response, IgE, which is a hallmark of allergy. And we know that this is coming from a different cell type, classically referred to as TFH cells, although I'll mostly focus on these type 2 Th2 cells today. So my lab has been interested in understanding a transcriptional repressor, BLIMP1, for a really long time, and BLIMP1 is traditionally associated with dampening down effector T cell responses. And so when we became initially interested in understanding whether BLIMP had a role in the Th2 cells in driving allergic asthma, we hypothesized that by removing BLIMP from the T cells, we would actually get worse disease. And so we were able to collaborate with Anuradha Ray, who's an expert at the University of Pittsburgh in type 2 immunity and in allergic asthma. And she helped us set up these mouse models in my lab. Prior to this, I had not done any allergic asthma work, where we were able to basically sensitize mice through chronic inhaled house dust mite every day intranasally, followed by a series of rests and re-challenges, which robustly drives allergic asthma in this mouse model. And so we can do this in control mice, and this is what you get when you do histology. You can stain with this periodic acid shift stain and look at this pink mucus in the airway, as well as these purple cells, which is showing the infiltration of the immune cells into the lung airways. And this is very classically what an allergic asthma patient would look like. And we hypothesized that when we used a mouse model where we could specifically remove BLIMP1, this transcriptional repressor, only from the T cells, that we would get worse disease. And we were really surprised when we found that we actually had the opposite effect. We had complete protection. And this was an absolute shock. In fact, Anuradha called me and said, "I think you mixed up your mice." So we did not mix up the mice. But it turned out that the reason for this was because we had none of these type 2 T cells that go to the lung and drive disease. And so that became the next portion of my lab to understand specifically how this was happening. And so we wanted to ask sort of two very simple questions about this transcriptional repressor BLIMP1. When was it being turned on, and where was it being turned on? So when you start an immune response, it usually starts in the lymph nodes, which drain all the different tissues. And so we decided to start by using a couple of very basic immunology tools, where we were able to get a mouse model where all of the T cells could specifically respond to house dust mite, and a mouse model where we had a YFP protein that would report on all of the cells that expressed BLIMP1. And by combining these two mice together and then taking those T cells and adoptively transferring them into a host mouse, we could induce allergic asthma with the house dust mite and then specifically track with flow cytometry when does BLIMP1 come on and in which cell type, and does it come on in the lymph node or does it come on in the lung. And without going through all of the details of this flow cytometry, what we found is that we could see low levels amounts of BLIMP1 in the draining lymph node as early as 3 days after we started the house dust mite, and that by 5 days we saw a really large predominant response in the lung tissue. So I'm not going to go through the data because it's all immunology and actually quite boring to this audience, but suffice it to say we did a lot of experiments with temporal deletion of BLIMP1, and despite the fact that you see a lot of BLIMP1 in the lung at 5 days, it turned out that it was actually this very, very early time point where you see very little of it that it mattered. This is when BLIMP1 was initially driving that Th2 cell response, which you needed to then go to the lung and drive disease. So that had answered the when question of when does BLIMP1 come on, and part of the where question, which is in the draining lymph node. But we wanted to more specifically ask where in the draining lymph node does this happen, and this is where we started to turn to spatial transcriptomics. And so we did these initial experiments using the V1 chemistry. So in this setting, we were able to do immunofluorescence of our draining lymph nodes at day 3 or at day 5, and we could use antibodies that could track the T cells that we knew were responding to house dust mite, but we needed the transcriptome information to know whether they were these type 2 T cells or whether they were these other T cells that I mentioned that could help produce antibody. And so by using spatial transcriptomics, we were able, even with this very low resolution technology, discern that the TFH cells were all in the B cell follicles out here, while the Th2 cells were sort of more in the interzones between the B cell and the T cell zones. So that told us that these two types of cells were located in different places in the lymph node, but we wanted to understand why. And this is where we turned to computational approaches. So at the time, we were beginning to collaborate with Jishnu Das, who's a very talented computational immunologist at Pitt, and he had developed this machine learning technique called SLIDE, which stands for Significant Latent Factor Interaction Discovery and Exploration, which basically is a machine learning algorithm that takes high-dimensional data sets like single-cell RNA-seq, and you can ask, okay, I have an observable feature. In our case, the observable feature was the localization difference between these two different types of T cells. And I want to know what are the latent factors or hidden variables that are going to underlie that observable feature that I have. Youjin had run his algorithm on single cell, but he had not tried it yet on spatial and was quite eager to know how it would perform. And so I was very happy to give him my data and see what we could find. And what was interesting is that he found several significant latent factors that are shown here as these Zs. And within each of these latent factors, you have a series of genes that the algorithm tells you which of your two observable features, in our case our two types of T cells, that gene is associated with. So for example, Z12 has all of these genes associated with our Th2 cells that we know leave the lymph node and go to the lung, and all of the genes in that Z in that latent factor were genes associated with cell adhesion and migration, so this made a lot of sense. But we were really excited when we saw this latent factor, Z13, because we had this very interesting molecule, IL-2 receptor alpha, that was associated with our Th2 cells. So this is a cytokine that's well known to be involved in T cell survival, but it hadn't really been shown to be involved necessarily in localization or differentiation. So the next thing we wanted to do was see if can we actually localize IL-2, the cytokine that binds to IL-2 receptor alpha, within those lymph nodes at that early time point. So we did that using RNAscope and was able to localize IL-2 in these purple dots here. And then we were able to then draw a circle around each of these dots and count the number of type 2 T cells that were localized by these IL-2 positive spots. And so we were able to show that there was a statistically significant enrichment of these type 2 T cells that expressed our molecule BLIMP1 close by to these IL-2 positive cells, and that these cells were located in that TB border zone that I had mentioned previously. So basically, using spatial transcriptomics and SLIDE, we had been able to now pinpoint the specific where this type 2 response was initiating to allergic asthma, and it's early in these lymph nodes in these TB border zones. And so basically what we were able to demonstrate is that you breathe in this allergen and you basically form what we now call these IL-2 microniches. And these microniches are critical for driving the initial response for these Th2 cells that ultimately go on and drive allergic asthma. So that was really exciting for us as immunologists interested in allergic asthma, but it also really helped us understand the power of spatial transcriptomics combined with computational approaches. And of course, throughout the process of trying to publish this work, we also, of course, saw the revolution coming with spatial. And so we wanted to next ask, okay, can we find a more high-resolution way to identify our cells? And at the time, this paper came out that combined spatial at a slightly higher resolution, 10 microns with beads, combined with TCR sequencing. And so we thought, okay, now we can actually use this to find our antigen-specific cells with our TCR sequencing combined with the transcriptome information, which allows us now to ask more refined questions. And so as we began to analyze these data sets where we could now better resolve our T cells of interest and the type of T cell that they were, and this work was done by my student Yizha Chen, we then also again started to work with Youjin on potential mechanisms to find ways to ask new questions about these types of data sets. And this work was also done collaboratively with Youjin Lee, who's one of our later speakers in the session. And we basically, Youjin and Youjin came up with this really fantastic computational approach called Space Traveler, which is an in silico perturbation method using spatial transcriptomics that allows you to identify functional microniches. And so using this data set that we had generated, they wanted to basically ask whether we could make sort of in silico perturbations, so in silico perturbing transcription factors or ligand receptor pairs inside the spatial space, and asking what are the downstream consequences of that in silico. So using Space Traveler, you can basically in silico perturb an individual transcription factor and see how it changes its localization in space, or how it changes its actual differentiation status, and how that changes the spatial architecture itself. So we used this combined TCR sequencing together with spatial transcriptomics data set that I showed you earlier to run Space Traveler and ask what are the key ligand receptor pairs that underlie this localization of these Th2 cells at this TB border zone. And some of the targets that came up, of course, IL-2 and IL-2 receptor alpha, which was quite nice because we had just shown in a paper that that was a key interaction. But we also were able to identify this interesting interaction between CCR4 and CCL5. So CCR4 has been well known in type 2 T cells to be the chemokine receptor that allows T cells to leave the lymph node and go to the lung, but it had not been previously associated with localization within the lymph node. So what was particularly exciting about this was that we had the opportunity to actually experimentally validate this. So what the Space Traveler algorithm was telling us was that these cells, once they became CCR4 deficient, these T cells would, rather than stay in the T cell B cell border zone, they would actually migrate closer to the B cell follicle. And that inhibited them also from migrating to the lungs. So we asked if we could actually functionally show that with experimental validation by CRISPR deleting CCR4 in our allergen-specific T cells prior to transfer, and then running spatial transcriptomics on those lymph nodes to see, can we actually see what we expect? So this is the outcome of that experimental validation using Visium HD. You can see that our control cells are sort of mixed throughout the T cell zones, but as predicted by Space Traveler, when you knock out CCR4 from our T cells specifically, you get the cells localized up against the B cell border zone, suggesting that these cells are now moving towards that B cell follicle space. And this is showing sort of the simulated knockout side by side with the experimental knockout. And this is finally just showing some statistical mean relative changes of these two things. So we're pretty excited about this as a computational approach because I think everyone wants to understand in their spatial transcriptomics data where are the functional micro-niches, where are the functional interactions that are occurring. But it's really hard just with that one static image in time to fully understand which of those interactions are important and which of those interactions are not important. With something like Space Traveler, it gives you the opportunity to in silico perturb those cells, those transcription factors, those ligand receptor pairs, and shorten the list of potential options that you might want to experimentally validate and can sort of actually go after the interactions in your spatial space that are potentially playing an important role in the biology. So I hope that that talk gave you a little bit of a sense of how we're doing things as a core and as also in my lab. I just want to end by thanking first my lab. So the initial spatial work was done together with Kunhi, who was a postdoc in my lab. He's now actually started spatial transcriptomics core at NIH for NIAID. And Will, who is the associate director of my core. And this is my lab, working on all different kinds of really cool projects. And of course, we have a lot of wonderful collaborators. And I also want to acknowledge my core facility and the amazing work that they've done, not just for the spatial transcriptomics in my lab, but also for the entire University of Pittsburgh. And with that, I'm happy to take any questions. And please come up to the mic if you do have questions.
Hi, thank you for the nice talk. I just wanted to find out a little bit more about your core setup. We have a fairly similar core we're starting at the Rutgers Cancer Institute.
Mm-hmm.
But we struggle with pipelines and the bioinformaticians that are the bottleneck right now.
Yeah.
So what are these basic analysis pipelines that you have at your core? What do you offer? And what goes on to the,
we basically just run Space Ranger, right? Okay. Because we've basically focused only on 10x. We were just running Space Ranger and sending the outputs of Space Ranger along with the FASTQ files to people, and that's in line with sort of our core in general doesn't do bioinformatics. Okay. What we have been doing is sort of trying to get a sense from people, what is the specific questions that they have, what type of tissue. For example, we have a cancer center, they have their own bioinformatics core, so if it's a cancer tissue that comes to us, we refer to them to that core. We have a core at Children's Hospital where the bioinformatics person in that core has really taken the time to learn all these spatial technologies so that we can refer things to her directly, and so she's been able to take a lot of the projects. And then when people have really unique computational approaches, sometimes I'll contact Jishnu and send them their way. It depends on whether I think it's a project that's going to really dovetail well with a particular scientist's individual interests like Jishnu. or with Youjin, or if I think it's a little bit more standard and can go off to more of a bioinformatic core. But we really, in particular, ask the customer to please set up a meeting with the bioinformatician even prior to starting the project. I think that makes a huge difference. And then to continue to meet with the bioinformatician throughout the duration of the data analysis. I think a lot of people think they're just going to email the data and wait for the Nature paper to come back to them. And that is not realistic. I'm sure many of you know that. So that conversation needs to be have all like throughout the process. Otherwise, it fails.
You need the bioinformatician at the project design stage.
Yes,
Ideally.
it helps. Yes,
Helps.
yes.
Thank you.
Yeah. I bet you guys are all questioned out at this point of the meeting, right? Okay, no worry. Oh yeah.
So for Visium HD, were you able to use the 2-micron resolution?
Yes.
So the result that we see was the result of 2-micron segmentation?
Yes, this data here is 2 microns. Yes, correct. So right now we're just, I didn't have the time to include this, and we're also not complete with the analysis, but we just recently compared the 3' Visium HD with the TCR sequencing to Visium HD with immunofluorescence. So that, because for me in particular, I'm very interested in trying to identify individual T cells that are individually specific to house dust mite and understanding the transcriptome of those cells and the niche that they're in. That has turned out to be a little bit trickier than we expected. Doing the Visium HD plus IF took a fair amount of optimization, right, Yvonne? But we got there. And then the 3' plus TCR sequencing, that was a little bit more cut and dry in terms of getting it to work. Our first pass on the analysis is that the gene capture between 3', as you would expect, and the probe-based Visium HD is not as comparable as we'd hoped. With 3', we got much fewer genes per spot than you get with the probe-based assay. So we are currently still determining which of the two technologies we're going to go for in the long run. Mm-hmm. Could be. I will tell you that lymph nodes are a pain to work with. You probably, if you don't already know that. They seem like they'd be so easy, but the cells are so packed in there. In general, like doing 3', like single cell RNA-seq from T cells is dead easy. It's not a challenge. So it's not totally clear to me whether we didn't quite get it right or whether that there's just a difference between the two. I've tried to ask a few people who are trying to also do the comparisons. And most people are just at that point where they're trying to make the assessment, so I think it's a little bit to be determined. But to be honest, I wasn't terribly surprised that there would be a difference, yeah. Okay, great, thank you. I'm going to switch gears to becoming session chair and introduce Andreas. You might want to put the little laser pointer on there. So, let's see. Thank you. So our next speaker is Dr. Andreas Pfenning. So Andreas is an associate professor in computational biology in the School of Computer Science and a member of the Neuroscience Institute at Carnegie Mellon University. He is a Sloan Foundation Research Fellow. He's received awards from the NSF, the American Federation for Aging, and his lab studies human brain disorders, where he is a leader in understanding the genetic influences of brain diseases using cutting-edge tools in genomics and AI approaches to understand how genetic differences in a population contribute to variation in human disease. So with that, I will let Andreas take it away.
Okay, thanks for that kind introduction and the opportunity to speak here. So I actually thought that the last question about, bioinformatics challenges was interesting. So, my primary appointment and my background is in computer science, and I'm within a school of computer science. And you hear a lot about people going in one direction, right? So, labs that are largely experimental, getting a ton of data and realize that they need a bioinformatician or a bioinformatics core to work through it. I'm actually moving in the opposite direction, right? Where even though I'm primarily trained as a computer scientist, I've been kind of slowly, sometimes rapidly building out the experimental infrastructure of my lab. And I feel pretty strongly actually that side is really important too. And ultimately what I think is the future is really a tight integration of the experimental technology with new computational approaches, especially AI, to solve some of the challenges out there. And that if you're able to merge the two, you're able to solve problems that you couldn't solve with either one individually. And so I also just want to make a note about Carnegie Mellon, my home institution. So we are not nearly as big as the University of Pittsburgh. And I don't know how many other Carnegie Mellon professors are here. We don't really have core facilities in the traditional sense. So basically, when somebody at CMU wants to do single-cell transcriptomics, they come to my lab. When they want to do spatial transcriptomics, they also come to me. When they want somebody to build AAVs, they also come to me. When they want any high-throughput sequencing done, they also come to me. I also have the only qPCR machine in the building for about a year or so we got a lot of people that way too. But actually, what's been really revolutionary is the idea that you're actually compensated for performing these functions at the university. I'm going to need to talk to the administration about finding a way to do that too. But so I want to actually talk today, this is a session on spatial transcriptomics about some technology that we've been developing there in that area. And so first I want to acknowledge my lab up in the top left there. A lot of this is work done by an MD PhD student and computational biologist, Mike Leone, who's down here, and then Robert Vanderweerd, a molecular biology and imaging technology specialist there, in collaboration with Becky Seal's lab at the University of Pittsburgh. So my lab, we have a lot of broad interests. So we build what are called sequence to function models in the computational biology or bioinformatics space, including developing new algorithms to do so. So if you take a piece of DNA or a genome sequence, we develop methods to predict its function. We also work on experimental genomic technology development. Initially, our focus was on methods to isolate rare neuron subtypes from tissue. And we also work on machine learning for comparative genomics too, working on a project called the Vertebrate Genome Project to sequence and analyze the genomes essentially of all vertebrate species on Earth, although we're only on the first couple hundred at this point. All right, so as Amanda mentioned, our focus is also in neurobiology, where cell type heterogeneity is a really big challenge, right? So even a small piece of brain tissue can contain hundreds of different subtypes of neurons, and basically each of these neurons perform different roles in the context of different neural circuits, different behaviors, different systems. And so disentangling that technology is especially important. And so I'll first talk about some Illumina sequencing-based in vivo barcoding technologies we developed. Really only talk about that briefly, but then move on to some of the Xenium, adapting that technology to Xenium spatial transcriptomics and some of our applications. Okay, so initially we developed this technology to understand the genetic basis of disease, in particular Alzheimer's disease. So this is a Manhattan plot for Alzheimer's disease genetics. Essentially, every skyscraper in the Manhattan plot is a locus associated with Alzheimer's disease, but it's still an open challenge about how we can interpret the individual nucleotides and the genetic variants that are contributing. Now, one of my major projects as a postdoc was actually showing that these genetic variants actually showed a tendency to impact the immune system rather than neurons, as a lot of people had been studying or had been assumed. So we wanted to know essentially to be able to measure and predict in a high throughput way what the impact of a given variant would be on neurons or be on the immune system. And that's where barcoding technology comes in handy. So the basis of the technology is called MPRA, or massively parallel reporter assays. So the basics of this technology is basically that you synthesize a regulatory element that you're interested in studying the function of, and you put that a regulatory element in front of a reporter gene, often a fluorescent reporter gene, but then you add on a unique barcode in the end. And basically, the more active the regulatory element is, the more of the barcode there'll be. So it's basically a high throughput or multiplex luciferase assay. And people have been doing these in vitro basically since around 2012. Now, a group had done this for Alzheimer's disease in vitro in HEK cells, but some of the results were a little bit puzzling, in part because HEK cells don't actually have a lot to do with Alzheimer's disease. So we wanted to be able to study the context-specific effects of these particular variants. So we basically developed a version of this assay that can be run both in vivo and in vitro. So we essentially have our cis-regulatory element, we pair that with a minimal promoter, and it drives the expression of a reporter gene and a barcode. And we used, in this case, AAV technology to be able to get large amounts of this library into the mouse brain to measure the function. So basically, for one of these plasmid libraries with the regulatory elements and the barcodes, we can in parallel measure the regulatory function of these sequences in the mouse brain, but also in vitro, in this case in cultured monocyte cells. And what we found is interesting and exciting, but also what we expect. So basically, similar cell types are highly correlated in terms of how active the regulatory elements are. But if you have cell types that are unrelated to each other, like for example, HEK cells and in vivo brain tissue, you basically get a very poor correlation. So a regulatory element that's active in the brain isn't especially likely to have high activity in HEK cells. Right, so I'm going to go through this a little bit quickly, but basically, in parallel to developing this experimental approach, we've been working on those sequence to function models that can take the sequence of the regulatory element and then predict whether it's active in HEK cells or brain tissue or the THP1 monocytes. And we're able to find some concordance between the models and even start to interpret exactly what transcription factor binding sites are involved and how these Alzheimer's-associated genetic variants might impact the function of these regulatory elements. All right, so that was the initial technology that we developed, and there's a preprint on that available. Now we've basically been moving on to spatial transcriptomics, and I'll switch domains from Alzheimer's disease to chronic pain. So for the chronic pain project, we're focused on the spinal cord, which is a major hub of chronic pain signals. And right now, the dominant therapeutic for, let's say, severe pain, or at least one of them, is basically a spinal cord stimulation, right, where you essentially stimulate the cells in the spinal cord. That disrupts their activity and you feel less pain. But there are a couple of major problems with this. One is that the technology is really invasive. The second major problem is that it's not especially specific. So if you stimulate too strongly, right, then you end up disrupting the function of a lot of different cells, and you can end up with tingling, your normal touch or sensation is affected. Sometimes you can even get an impact in movement. On the other hand, if you don't stimulate strongly enough, you're not actually going to impact chronic pain. So we wanted to develop a therapeutic that was actually targeted to the cells involved in chronic pain, but that leave the other cells in the spinal cord intact. All right, so the first thing that we had to do, and we started this project 5 or so years ago before there were a lot of single cell pipelines out there, but that's one of the advantages of being a computational biologist. We developed a lot of these from scratch, or essentially used some barebone tools to be able to do this initially. But we were involved in building one of the first molecular maps of the spinal cord, in particular the primate spinal cord in rhesus macaque. So we identified these different populations of cells, different cell subtypes, and we've been working, and I'm just summarizing a ton of research over years, working with our collaborator Becky Seal at the University of Pittsburgh to basically figure out how these molecularly defined cell types map onto the cell types that people have been studying in the context of chronic pain. So part of that initial atlas was basically doing some spatial transcriptomics to look at the spatial localization of the different cell types. And this is a little bit of an aside, but I think it's an important point, so I want to make it. Some of the original tools for analyzing the spatial transcriptomics and the single cell didn't work especially well. In fact, data sets that were gathered by different labs were disagreeing about what cell types were present. And what the spatial transcriptomics revealed is basically that there are 2 different axes of cellular molecular identity. One of them that you could see with the spatial transcriptomics was anatomical. So as you move dorsal to ventral, you have gradients of gene expression that influence the molecular profile of the cell. Orthogonally, you also have different developmental trajectories that are independently influencing the identity of the cell and the transcripts that are expressed. And without accounting for both the developmental hierarchy and the spatial information, we were getting conflicting or incomplete pieces of information. I'm going to gloss over this a little bit, but we did find and confirm that certain cell types were highly enriched for genetic variants associated with the predisposition to chronic pain. So that was essentially a hint that we were going in the right direction. And then again, harmonizing this with information from Becky Seal and detailed work that she did with transgenic animals and other techniques, we're able to work out how the cell types we were identifying map onto the circuitry of the spinal cord. But the next thing we wanted to do is not just understand which cells were associated with chronic pain, but actually be able to target them. And for that, we decided to use AAV, or adeno-associated virus. So people have been working on methods for high throughput screening, improving capsids for AAVs that are able to make this technology much more efficient. Unfortunately, that hasn't really produced things that are super targeted, at least when you're dealing with one very specific subtype of neuron versus a group of others. But what we wanted is basically a high throughput screening of promoters to be able to know which promoter would activate the cargo in the specific cell types involved in chronic pain. And this is essentially a part of a larger approach, again, that people have mostly been doing in vitro to iterate the genomic identifications of the regulatory elements with artificial intelligence to be able to design sequences that will only activate in the cell type of interest, but not activate in any other cell type. So the overall pipeline we put together is where we start off with a whole bunch of information, including our single cell experiments and experiments that have been made publicly available, and we put that into an interface, identify what cell types are present, and also get the transcriptomic and open chromatin information from these cell types. We then use artificial intelligence approaches, including some designed in our lab, to both prioritize promoters and design synthetic ones that are only going to activate in the cell types of interest. Then we move on to spatial transcriptomics, and I'll talk about that technology, and use similar barcoding approaches to validate whether the promoters that we design are actually specific for the cell types related to chronic pain. And then we actually have turned this into more of an iterative loop where we can then use that information to go back in and refine our hypotheses about what sequences and what sequence features are going to be specific to a cell type. So maybe in slightly more detail, we start off with single cell open chromatin data. That serves as input to these sequence to function models that can prioritize or predict cell type specific promoters. We then package those into AAV, allow it to drive GFP, and then traditionally a lot of people, including my lab, have been using those fluorescent signals to be able to identify, all right, is this expressed in the cell type of interest? And again, this is a little bit of an aside, but we've already successfully used this approach more on the AI side to actually be able to design cell type specific tools that are functional in the non-human primate brain, in this case used to drive channel rhodopsin and selectively stimulate neurons that are in layer 3 of the rhesus macaque brain. So we first started out in terms of the computational side by taking the single cell open chromatin in mouse and in macaque and training machine learning models to predict which regions would function as cell type specific promoters. We then started using spatial transcriptomics to validate the individual enhancers. So we can measure the endogenous genes to get the cell type identity. So for example, we have the EGFP here that's driven by the enhancer at the same time to say, okay, are we targeting the cell types that we hope? Okay, now this approach of basically screening these enhancers one at a time has been somewhat successful. But basically, if you are involved in core facility research, you can probably understand how time-consuming that is. Now, one example of that is the work that's being done by the Allen Institute that's doing this. So basically, they clone the enhancer into this AAV-based reporter. They package it up, deliver it retroorbitally, section the brain, and then one at a time, look at the cell type specificity of these enhancers. Now, overall, there are no functional issues in that it works, but there are some conceptual issues. So this is one publication that identified 7 or 8 cell type specific enhancers, and you can see what the author list was and what the average cost was per enhancer. And there's another publication here where they identify another handful of enhancers, again, enormous author list because they basically had to go through and validate these one at a time. And this is a summary of the tools that they were able to identify. So what we wanted to do is basically do this in a much more high throughput way. So this is overall how our library construction works. So we basically have the cis-regulatory element or the enhancer promoter here. We use a nested cloning strategy to insert a reporter, a fluorescent reporter here with a synthetic intron for stabilization. And then we have a barcode here on the 3' end that allows us to map the enhancer activity. So basically each regulatory element is associated with a unique barcode. And so we basically take this now library of regulatory elements, controlling the synthetic barcodes, and then we use the AAV technology to get this entire library into the mouse spinal cord. So the library was pool injected. Our collaborators in the SEAL lab dissect the spinal cord, prep the tissue, section it, and then we put it on the Xenium to look at the spatial transcriptomics. And I'm running short on time, so I'm just going to summarize. One of the really insidious challenges here is basically deconvolution because of some technical artifacts that can occur when you're working with AAVs. So in other words, when you have AAVs, you can have the regulatory element on one of your constructs actually regulating the barcode on another construct because they chain together. And this had been confounding other uses of the technology throughout the years, other people who have tried to do something similar. The Xenium is actually sensitive enough that we can pick up basically each individual instance of a barcode being expressed within a given cell type, and that allows us to perform a deconvolution step. So after deconvolution, we've been able to get positive controls. So for example, before deconvolution, we see off-target effects with HSYN, which should only be active in neurons. And this is actually a barcode that was designed for motor neuron enhancers. And again, it showed a ton of off-target effects until the deconvolution was done. We also include spatial transcriptomic deconvolution as well to take the spatial information into account as we're performing that step. And after that, we were able to get good concordance of the controls. So in other words, known neuronal enhancers were neuronal, and known oligodendrocyte enhancers are ones that we predicted would be oligodendrocyte related. And we also use this to validate promoters that would selectively activate in the specific neuron subtypes we predict are related to chronic pain. We then use those to drive chemogenetic tools, which, to give you a very quick neuroscience primer, shut down all of the activity of the neurons that they're expressed in. So it was a way to take the neurons involved in chronic pain and shut down their activity. And what we found was really encouraging. So basically, in a mouse model for chronic pain, our collaborator Becky Seal found that when you shut off these neuron subtypes involved in chronic pain, that the mice were no longer sensitive to one form, mechanical allodynia, but normal touch or sensation was preserved. They've subsequently gone in and done a bunch of other experiments for different types of chronic pain, including inflammatory injury, polyneuropathic or diabetic pain, and neuropathic injury or essentially nerve injury pain. And in each of those cases, selectively inhibiting these cells was able to improve symptoms of chronic pain while keeping normal touch, sensation, and movement completely intact. So again, this is a way in which that we've been able to use a basic analysis of the single cell data, AI tools to design synthetic enhancers and promoters, and then spatial transcriptomics basically in a loop to be able to design these tools to selectively inhibit specific cell types and then have preclinical evidence that it is safe and effective at inhibiting chronic pain. We're now working on, of course, a number of different applications, but I think this is probably a good place to wrap up. So I'll take any questions you might have.
So I'll go ahead and get started. That was really fantastic. How easy would it be to transition this framework with the AI designed synthetic enhancers and these libraries to another tissue? Like, for example, if you want to do this in like the liver, for example.
That's a good question. I mean, they're like, we can, so the first time we did this, of course, it took years, right? And then the second time we did it, it took more like months. So we're still working through the kinks and trying to identify the pain points and make it faster. One of kind of the nuances is it actually relies heavily on cloning and effective cloning, right? If you don't clone your library in a balanced way, you end up measuring 3 enhancers predominantly, and then the other like 30 enhancers you can barely measure. So there are a lot of different nuances and steps to this, but we're in the process of trying to make it more reproducible. There isn't any reason that something like this couldn't be run in another tissue as long as you have the expertise. In fact, a former postdoc of mine is now running her own lab and running some of these technologies in liver itself.
Okay.
And she's having some success there. The AI side too, we're also working on ways to make that more accessible. So for example, I teach a class to primarily biological science and neuroscience majors that have only had a semester of programming. And I've been working with the staff in that course to basically create an interface to make it easy to train those models. And in that case, the students are actually able to do it relatively well, right? As long as, you know, they have a little bit of programming expertise, we teach them about the biological intuition, and then they're able to kind of understand how, oh, like, I need greater specificity, so I'm going to tweak this parameter. So not all of it is completely automated, but we're working on making each of these parts easier to adopt.
Okay. And then my second question is, how challenging was it to design the probes for Xenium for each of the barcodes for the MPRA?
Right. So this is something that we worked with 10x Technologies on. Now, the downside is that they tend to focus on like, oh, you want to measure this gene, give us the sequence. And, you know, of course, we're working with synthetic barcodes, so there aren't really any predefined tools to do that. But on the positive side, there's actually flexibility at 2 levels, right? We can use almost any synthetic barcode as long as it doesn't have enhancer activity itself. So we can be flexible, and if one barcode of ours doesn't work, 10x can tell us, oh, hey, your GC content is off, or there's a region of low complexity. Do you think you could make a new barcode? And it's easy for us to do. I will say in the first iteration of the technology, we used roughly 90 different barcodes. And then there were like 4 or 5 that had off-target effects. So in other words, when we ran it on tissue that had no library, we still picked up some signal. So I think there's still a little bit of iteration to be done. But given that these are 500 base pairs long, I'm actually very happy so far with the amount of specificity and the reliability we've been able to get.
Yeah, great. Thank you. Anybody else?
All right, thank you.
I have to keep going back down there because I can't see the slides from up here, and I want to see the talks too. Okay, so our last speaker is Dr. Youjin Lee. So Youjin is actually my lab neighbor. She is an assistant professor of immunology and also a member of the Center for Systems Immunology in the Tumor Microenvironment Center at the University of Pittsburgh. After receiving her PhD from Harvard, she trained with Dr. Alex Marson at UCSF, where she developed XYZ-seq, which is a single-cell spatial transcriptomics platform. She was also a Parker Bridge Fellow in the Parker Institute for Cancer Immunotherapy before joining us at the University of Pittsburgh a couple years ago. Youjin is a leader in spatial and systems biology as well as in functional genomics, and I'm excited to have her talk. So take it away.
Great. Thank you. Okay, great. Thanks, Amanda, for the introduction, and just want to say that I'm actually very different from the previous 2 speakers. Amanda obviously runs the sequencing core. Andreas seems like he's a one-man show at Carnegie Mellon running his own core. I am simply just an assistant professor trying to survive. So today but I think I know why I'm here, which is that I have a novel platform for spatial sequencing that I'd love to share with you guys. So the title of my talk today is, I'm gonna basically present a novel spatial multi-omics platform that can integrate spatial labeling directly to single cells And at the same time, I'm going to try to give you a little bit of a little expert on how CRISPR perturbation workflows can also be integrated to that workflow. Okay, here's my disclosures. Okay, so what do we do? So at a high level, my lab essentially tries to build tools to try to understand the cell-cell communication networks within the tissue microenvironment that drives disease. And to answer this, we actually try to build wet lab tools or experimental tools, specifically for spatial transcriptomics, as well as machine learning tools. But initially, like everybody else, we started with single-cell RNA sequencing to try to identify various cell types, function, and cell states. But as all of you can probably appreciate for this audience, the moment you try to generate samples for single-cell RNA sequencing, the entire spatial context is destroyed. So it's very hard to study cell-cell communication when you actually do not have any markers to understand how they are working within each other. So we turn to spatial transcriptomics, and as a lot of people have already talked about, as well as the audience, that there are many different types of commercially available spatial transcriptomics platforms. And I would say they kind of fall into three broad categories. One is kind of the capture-based system, where you end up having a oligonucleotide that is then placed on top of the slide, tissue is laid on top of it, and then the RNA is captured and sequenced for spatial resolute data. And then the second method is the image-based method, where you have a probe that is then specific for a particular gene of interest, which is then hybridized and imaged using various combinatorial barcodes, and then to identify various transcripts that are directly in the cells. And then there is the third method, which is the in situ-based sequencing approach, which is a whole transcriptome-based, but at the same time, you end up having in situ-based reverse transcription that occurs, and then spatially resolved for sequencing at the later end. But there are various trade-offs to the spatial sequencing platforms. The way I see it is essentially two ways. One is either you get scalability, meaning you get the whole transcriptome, many different tissues at the same time, a lot of data, or you get the spatial resolution at a fine resolution, at almost at a single cell resolution itself. And then that is with the image basing as well as in situ-based sequencing approaches. So what I did was, during my postdoc, I actually developed this platform called XYZ, which is the in situ-based sequencing platform. And as the name suggests, it is XYZ spatial coordinates mixed with sequencing that links spatial metadata with single-cell RNA sequencing. And then, just like most platforms, we have now gone into version 2, which is a single-cell spatial multi-omics that allows for true spatial resolution single-cell data. And that's what I'm going to be talking to you about today for the most part. So the way the methodology works is pretty simple. It is that we have these oligos that are deposited into these microwells, and then we cut a slice of tissue, lay it on top of the array, and then the spatial oligos get transferred onto the tissue. It goes through normal single-cell RNA sequencing pipeline where the tissue is digested, and then it gets funneled into various commercially available platforms like 10x. And because of the fact that 10x currently, for example, allows for single cell to be paired with various multi-omics platforms, this platform actually allows for you to integrate spatial tagged cells with spatial single cell RNA sequencing that is also multi-omics. Okay, so the first part I wanted to show you is the fact that you get really good quality data. Here you have on the lower right corner is the tissue laid on the array. And as you can see here from the spleen, you can get all the different types of cell population that are associated with the spleen. And for example, dendritic cells or various T cells. But because the quality of the data is so great, you're able to now even fine-tune and subtype the various DCs versus splenic, migratory, and myeloid, as well as effector cells or helper T cells that are found within the population. And as a result, as you can see here, we're able to integrate multi-omics, spatial multi-omics to be exact. You get the RNA expression. This is in fact a liver that has been injected with tumor. And then not only do you get the RNA expression, the various UMAPs and all the different populations, including the hepatocytes and the tumor. But as you can see here, because of the fact that we can then generate multi-omics data, we're able to also see the protein expression. Here you have CD45 or integrin A1 that was also captured at protein level. And at the same time, because each of these cells are spatially tagged, you can then map them back into the spatial location where they originated from. Here you have the spatial organization of the various cells that were found in the tissue. Okay, so here we end up having a different type of, as I mentioned earlier in the title, that I'm going to show you sort of how these multi-omics can be incorporated. In this scenario, you have not of any sort of spatial or RNA or protein capture, but you actually get the guides that are captured within the tissue. Here you have 4 spleens that were spatially sequenced with XYZ version 2. And then you can see that various guides or genes that were edited can be captured in various tissue localization within the spleen. So these are 2 different types of genes that were captured by guides, and as you can see, what is really interesting is the fact that they actually localize in various populations within the spleen microenvironment. So for this audience, I wanted to also kind of show you what the technology kind of looks like and how the array is shaped. As you can see here, this is sort of the device where it's instrument-free. You have the microarray that is then spotted with the various spatial oligos. The array that we currently have is by 10 millimeter by 20 millimeter. And then we have the 2 different types of chemistry that we can actually apply to this particular technology. And as a result, we're actually able to do both fresh tissue. This is by the antibody-based system that I developed during my postdoc. And then we have this thing called the multi-seq, which then allows for us to tag FFP or frozen tissue as well. That allows for nuclei profiling. And as I mentioned, it's able to dovetail really directly with the existing core facilities, single-cell RNA sequencing capability. We already know that it's able to work really well with the 10x genomics, obviously validated with Illumina sequencing. It's able to generate compatible data with TotalSeq, with BioLegend, which allows us to do protein, and then the 5' as well as the CRISPR. So all these compatibility allows us to basically generate a single-cell spatial multi-omics platform that allows us to not only get the data, but there's another unique feature associated with this, in a sense that because of the fact that we're able to dissociate the cells once they have been spatially tagged, we're able to generate an enrichment. So what that means is that because there's a fact sorting step prior to popping into the 10x platform, you have, for example, here an example where there's no enrichment. You have mostly hepatocytes in the liver. But then you can then also sort for non-hepatocytes, and then getting all the other, or hepatocytes enriched, and then you get all the other depletion of the other liver cells. And then you can also get a selective enrichment of the macrophages that are also present as well. So this unique ability allows us to, for example, take spatially tagged cells that are enriched for the population of interest that you would like to sequence. There's also various type of sample compatibility. So there's multiple tissue types and cells that you can actually assay with this. We've showcased, as I mentioned, the mouse spleen. We can also do the kidney, but as well as the mouse brain as well. And we've actually expanded this to the lung, skin, and obviously the lymphoid tissue. Okay, so for the last few parts, I'm going to actually show you the applicability of utilizing spatial data. So here you have a mouse kidney that we actually generated spatial sequencing data from using XYZ2. And as you can see here, you're able to see the various cell types that are found within the kidney. One of which was actually the myeloid population that we were able to subgroup as a MAC0 or MAC2. 2 different subtypes that were found. But because of the fact that every single cell has been spatially tagged, we can remap them back into the original location. And when we did that, what we're able to find is that, for example, there's a spatial distribution differences between these 2 populations, which you would have not been able to understand or get if you were just to do single-cell RNA sequencing. So for example, here you have MAC2 that is co-localizing in the outer region of the kidney, and then you have MAC0 that is co-localizing in the inner core. And this is actually a component that Amanda has already presented earlier, but we did work in collaboration with, I worked in collaboration with Youjin and Amanda for this. So here, as she mentioned, that we have this thing called Space Traveler, which is a computational model that essentially combines spatial transcriptomics with CRISPR perturbation. And then so what we're able to understand is when we train on spatial data, you're able to not only understand how the cells are impacted intrinsically, but then because of the fact that you're training on the spatial data, you can kind of understand how the cells are co-localizing or cell communicating with each other. So as a result, when you're able to computationally perturb a gene by the cell, you can actually not only understand the impact that it has on the cell, but at the same time, how it propagates across the entire tissue space. So thankfully, she didn't present this data, but this was our kind of our proof of concept sanity check that it was working. What we have here is spatial transcriptomics data from the human tonsil. And as you can see here, you can see the various populations that are usually found. And then because this is spatial data, you can then do a 2D spatial map of the various cell populations that are found within that tissue space. And through various computational methods, we ended up kind of focusing on germinal center B cells that, as you can see here, is highlighted in this blue region and outlined by black. We found this gene, transcription factor FOXO1, that is highly expressed in this germinal center B cell population. And then so when we computationally knocked it out, what we found was that it seems to predict, the model predicted that if you're to knock this out, FOXO1 in germinal center B cells, before knockout you have not only the orange, the dark zone germinal centers, but then you have the light zone germinal centers at the bottom. But after the FOXO1 knockout, what you end up seeing is a abrogation of the dark zone germinal centers, which then reverts to majority light zone germinal center B cells. But what is really interesting to us is that when we then, although the computation kind of simulated what the impact would be, when we dug into literature, we actually found that back in 2017, people had actually done this, where when you knock out germinal center, FOXO1 or germinal center B cells, it led to the developmental loss of the dark zone germinal center B cells and the disruption of the overall germinal center architecture. So this to us proved to us that our model was in fact working, and then obviously we applied that to Amanda's model to try to understand how it would actually impact disease in the allergic asthma model. And with that, I am going to end my talk. Thank you so much. Happy to take any questions.
So I don't think you mentioned, but how big are the wells for XYZ?
Sorry. So the wells currently is about 80 micron, but we can go down to 50 at the moment, so 50 to 80 micron.
And then if I recall correctly, you need some computational approaches to still arrange the cells within that 50 micron space. Is that correct?
Actually, you don't.
You don't?
So yeah, because it's actually true single cell data that you generate within that each well. And so as a result, you don't really need to figure out how they're arranged in space.
Oh, okay. And then remind me, so the top layer of the tissue gets the spatial tag.
Yes.
If you wanted to do multiple sections, would you be able to do that? Can you do the top layer and then cut it and then do it again?
Yeah, so you can essentially take sequential slices, and then each time you can then barcode each slice, and then sequence everything to get the whole Z layer or Z stack.
Okay, thanks. Any other questions?
Is it compatible with IF or H&E staining?
So what we do currently is you cut the tissue slice in half, and then on one side what we do is we do spatial sequencing, and on the other side we can do H&E to kind of get the overall structure and the architecture of the tissue. Not on the same, but it's essentially kind of the mirror image of it. So if you think about like an apple that's cut in half, then you can get one side where spatially sequenced and the other half that is then imaged, just to kind of get the layout of it. But the quality of the data actually allows us to, you don't have to have issues with deconvolution or cell type segmentation. So as a result, you're able to generate really good bona fide data that allows us to determine what the cell type is within that region.
Do you know, is it Is your workflow compatible with those experiments that use live tissue slices?
Sorry?
Is this workflow compatible with those live tissue slice
Yes. You
experiments?
can do, in fact, that's sort of kind of the reason the quality of data is so great, because you're working with already fresh tissue to begin with.
Right.
So this essentially, if you think about it, is it's 10x data, so single cell data that people use that have been pre-labeled with spatial oligos that allow you to spatially map the cells back into tissue. So it is kind of, it works with any sort of single cell platform that currently exists, and then all you do is simply, you can now get additional data, which is the spatial data.
Have you tried it with live tissues, like brain slices and things?
Yes, those are all, the data that I actually showed you, they're all from live tissue. The multi-seq method that I mentioned earlier is actually the one that allows us to do it in the FFPE or frozen tissue slices.
So I think the workflow that we were struggling with a long time ago was live brain slices and then imaging for, testing with drugs, and then taking that live slice out of culture and into single cell.
Yes, but then you can absolutely do that.
And
You can do that,
with
exactly. So I think we showed, you can see the compatibility. So here's the mouse brain.
Brilliant, thank
Yeah.
you.
Yeah, very cool work. Thank you. So there's actually one thing I'm really curious about, right? I'm used to seeing data like, perturb-seq done in vitro, right? Where you have all of these different cells and you manipulate various genes and then look at the effects. But of course, with that, you don't have the spatial information. So I guess I'm curious, when you're doing these spatial perturbations, like what's the distribution of ranges in that, in that space that it affects, right? Do most perturbations just impact the individual cell, or maybe the individual cell and then a few around it? Or is it common to see like one perturbation impact entire sections of the tissue like FOXO1?
So that's actually a really interesting question. What we find is that the perturbation seems to also impact that cell itself, but then it also seems to impact the neighborhood. So it's exactly how we imagine. The beauty is that you're able to capture the guides in conjunction with getting the entire transcriptome of the cell. So you can actually see how a cell, the same exact cell that is perturbed in one location, and the exact same cell type that is in another location that's not perturbed. And you can kind of compare the differences of how it impacts intrinsically as it embeds within that local environment. So the edit actually modifies a cell to go to unique areas, is sort of what we're hypothesizing. But at the same time, what we do see is that we're seeing the differences in the area, the surrounding area, the neighborhood, per se, of the cells.
Okay, great, thanks.
Okay, thank you so much.
Okay, we're going to close the session. Thank you so much for everyone for coming. It sounds like the party's next door. I will refer you there.